Hello everyone, Mr. Linder here. Let's talk about the process of beta oxidation. So in previous videos, we've talked about how glucose, a carbohydrate, can be used to generate ATP. But you should also understand that we get energy from proteins, we get energy from fats. And so beta oxidation is an important process to understand because we generate a large amount of energy from lipids, from our fats. So it turns out that lipids essentially are a primary fuel storage molecule in the human body. And it's a primary fuel storage because of its high energy content. Compared to proteins and carbohydrates, fats actually generate 9 calories per gram. So 9 kcal or 9 large C calories per gram of fat. So for example, if you only had 2 grams of fat, you're talking about generating 18 calories of energy. And so lipids are an interesting molecule to discuss because of their high energy yield. In order to get energy from lipids, we need to be able to digest lipids. So on this particular diagram, this is just showing you a generalized idea of how we get energy from lipids, from triglycerides. So triglycerides, this would be a molecule with glycerol and 3 fatty acids. So you have to imagine there are fatty acid chains coming off of the glycerol. So 3 fatty acids off of the glycerol, that's a triglyceride molecule. We can use enzymes known as lipases. And lipases are going to separate the glycerol and the 3 fatty acids. And this process is known as lipolysis. So we use lipolysis in the digestive system, predominantly using something called pancreatic lipase in order to break down triglycerides. And then we're going to eventually absorb the products into our epithelial cells. There's some conversion reactions that take place, and then we move the lipids into circulation in the body. Glycerol that comes from the breakdown of these lipases can actually become a subunit in glycolysis. So if we were to zoom in on that, give you a little bit better view of what's going on, the glycerol that comes from the digestion of a triglyceride can actually enter into glycolysis. So it's going to enter in as a phosphoglyceraldehyde molecule. So go back and watch previous videos on glycolysis. So the glycerol molecule can actually be used to generate energy. We also, though, have the fatty acids that come from the digestion using lipases. Those fatty acids are going to go through the process that we refer to as beta oxidation. So let's take a further look at that. So once you have these fatty acids, those fatty acids are going to enter into the mitochondria. So this is the mitochondrial matrix, and in the mitochondria is where we see the process of beta oxidation taking place. Beta oxidation essentially is a process of using enzymes to remove 2 carbon subunits. So we're going to remove 2 carbon, essentially acetate subunits from a fatty acid. So when looking at a fatty acid, you'll notice that it's a long chain of these carbons. And so when you break off 2 carbons, you can use those 2 carbons, this acyl unit, as a means of entering into the citric acid cycle. We can create acetyl-CoA, and we can remove that coenzyme A, and then that acyl unit can fit into the citric acid cycle. And if we can insert these 2 carbon subunits into the citric acid cycle, this is similar to what we do with pyruvate. Pyruvate comes from glycolysis. We can use the transition reaction to generate acetyl-CoA, and it's the acetate units that enter into the citric acid cycle. So all we're doing in beta oxidation is we're creating these 2 carbon subunits that can find their way into the citric acid cycle. Since fatty acids are long chains of carbon, we can break off multiple 2 carbon subunits. So the first 2 carbons that come off from beta oxidation, we can use those for energy. The next 2 carbons that come off, we can use those for energy. The next 2 carbons, the next 2 carbons, the next 2 carbons. So beta oxidation is a very powerful process because we can keep generating these 2 carbon subunits, and we can keep inserting them into the citric acid cycle and thus essentially generate energy from the electron transport chain. We're going to get our high energy electron carriers. We're going to get our NADHs and our FADH too. We're going to run those through the electron transport system, and we're going to generate ATP from them. So thus, we can get a much larger yield of energy, that 9 calories of energy, compared to only getting 4 calories per gram for proteins and 4 calories per gram for carbohydrates. So how does this beta oxidation actually work? So let's take a look at the process of beta oxidation that's going to be taking place inside the mitochondrial matrix. So what you're looking at here is the reactions that are taking place in the mitochondria. So this is the mitochondrial matrix. And what's happening to that fatty acid, so this is that fatty acid molecule, this long chain of carbons. What's happening to that fatty acid is we're using enzymes to undergo this beta oxidation process in order to create those acetate units. Okay, so those acyl units, right, are coming from this beta oxidation process. So this is beta oxidation. And beta oxidation is just a series of reactions, okay? And so you can see these reactions here, okay? Step 1, step 2, step 3, step 4, step 5. You can see this series of reactions that allows us to generate these acyl units, and we refer to this as beta oxidation. This is enzymatically catalyzed in the mitochondrial matrix. There's essentially 4 main enzymes that are used in beta oxidation in order for us to shorten these fatty acids and essentially chop off 2 carbon subunits at a time. So if we look at this process inside the mitochondrial matrix, what do we actually see taking place? So in this first reaction, we actually see us using ATP. So we're actually going to use 1 ATP in the first part of this process. So that's actually a loss of energy, but we're going to get energy back as we go through this. In the next reaction, what you see is the generation of an FADH2 molecule. So we're going to generate one of these high energy molecules as we run this sort of second step of the reaction. So first step, we use up an ATP, but in the second step, we are getting 1 FADH2 molecule. In step number 3, we see the addition of water. So water is being used as we're changing the structure of the molecule. In step number 4, we see that we are generating one of these NADH molecules. So we generate 1 NADH, and we can put the plus H plus. Okay, so 1 NADH molecule. And then in the final step, we can see that we are generating this acetyl-CoA molecule, this acyl unit. And beta oxidation essentially is complete for that first process. We've removed a 2-carbon subunit, and then the acetyl-CoA is entering into the citric acid cycle, or what we call the Krebs cycle. When the acetate unit enters into the citric acid cycle, it's essentially binding with oxaloacetic acid to form the first molecule, citric acid, and then it's running through the cycle of reactions that we know as the citric acid cycle. Go ahead and go back and watch one of my citric acid cycle videos to look at the reactions. But from the citric acid cycle, from the Krebs cycle, what do we actually get from this? The Krebs cycle actually yields 1 ATP. It yields 3 NADH molecules, and it yields 1 FADH2 molecule. So if we were to look at what we actually get from beta oxidation, for every 2-carbon subunit that is removed, we're going to net 13 ATP. Okay, so for each, this is for each 2-carbon that's removed, for each 2 carbons. And remember, fatty acids typically have anywhere from roughly 16 to 18 carbon units. So you can generate a lot of energy from 1 fatty acid molecule. And that triglyceride had 3 fatty acids. So you can see why there's a much larger energy yield from fats, and why it's such a good molecule to store long-term for energy usage. So where does this 13 ATP number come from? Well, we know that we used 1 ATP. So we're going to come back and look at that. But let's take a look at what we get from just the process of beta oxidation. If we have 1 FADH2 and 1 NADH molecule, and those are going to run through the electron transport chain, when 1 FADH molecule donates its electrons, you get 1.5 ATPs. When an NADH molecule goes through the electron transport chain, you get 2.5 ATPs. So if we were just to add up what we get from beta oxidation, you're looking at 4 ATPs being generated, but we used 1 ATP, so we're going to minus 1 ATP. So there is 3 ATPs that we're going to net when we generate these high energy molecules from beta oxidation. We also have that acyl unit entering the Krebs cycle. So we generate an ATP, plus we got 3 NADHs and 1 FADHs, and those are going to run through, these guys are here, they're going to run through the electron transport chain as well. So 3 NADHs, okay, that's 3 times 2.5. So that's going to yield us, if we have 3 times 2.5, that's going to give us, what is that, 7.5 ATP. Okay. And then 1 FADH times 1.5, so that's going to give us 1.5 ATP. So if you add that up, that's 9 ATP molecules. And then, of course, we made 1 from the Krebs cycle. So 9 plus 1, that's 10 ATP that we're getting from the products of the citric acid cycle, or from the Krebs cycle. So we had 3 ATP that we net from beta oxidation. We have 10 ATP that we get from the products of the Krebs cycle. And so 10 plus 3 is our 13 ATP. So for every 2 carbons, we see 13 ATPs that are produced. So if we were to chop off another 2-carbon acyl unit, that's another plus 13 ATPs. So with just 2 of the 2-carbon acyl units, we're up to 26 ATP. Okay, so that's 26 ATP from just two carbon units. If we did a third acyl unit, that's another plus 13 ATP. So now we're up to, what is that? That would be 39 ATP, and so forth and so forth. So because a single fatty acid can produce so many of these acetate units, lipids essentially contain more energy than proteins or carbohydrates do. And then, of course, we get energy from the glycerol portion of the molecule. And so that's going to generate energy from the triglyceride molecule. And so we're seeing just a greater and greater ATP yield. If we have excess fatty acids, then we can convert those fatty acids into ketones. So you may be familiar with the fact that fatty acids can become ketones. Ketones are going to be molecules like acetone, acetoacetic acid, beta hydroxybutyric acid, and these are all molecules of ketones that actually can be used to supply the brain with energy if you had low glucose levels. But we also run the risk of generating these keto acids that could lead us to ketoacidosis. So we do have to be careful with our homeostatic balance. That's why ketogenic diets, diets that are high in fat, high in protein, and low carbs, can actually shift us into ketoacidosis, and pH balance can throw off homeostasis. So we do need to be aware that excess fats actually do lead to excess ketones, and that can change our pH situation in the blood. So that, though, is a little bit on why we store lipids and how we generate energy from lipids, either through glycerol or through these acyl subunits that come from fatty acids. So I hope that helps. Take care.